mean, most of you are, but some of you aren't. Okay. You can Google it, you know, but why spend the time if I'm just telling you now? Okay, so Campylobacter causes a diarrheal syndrome, and Neisseria can cause gonorrhea. Okay, so I'm telling you the answer here. You guys probably should be writing these down and doing it. Do it now. Don't try to do it over the break. That's ridiculous. You know, 2 weeks from now, you won't remember these conversations. Ooh. Number 4 has a little bit of vocabulary, a little bit of common sense, critical thinking. So, anybody know what a capnophile is? What do you know, Emma?
It like thrives in high carbon dioxide concentration.
Correct. Thank you. Capnophiles like CO2. Capnophiles love that. Phile or philic means they like something. Capnophile means they like CO2. So, why does it make sense that many human pathogens are capnophiles? It means bacteria that cause infections in human tissues like CO2. So this is just the common sense kind of. So, guys, remember in the last lecture exam, you had to know about aerobic respiration. Remember that? Because most cells of your body go through aerobic respiration. Aerobic respiration, what does it do? It takes in oxygen, blows off CO2. That CO2 is in your body most of the time. It has to get picked up by red blood cells and then brought back to the lungs. So your body is constantly producing CO2 when it breaks down molecules like glucose. So your body tissues have a lot of CO2 in it. And it makes sense that organisms that live there, they have to tolerate that or maybe even like that. Why does it make sense that most people that live in, you know, Newport, Dana Point, San Clemente, Laguna, why does it make sense that they live by, you know, the ocean? Oh, because they like the ocean, you know, they like it. So it's asking the question the other way around. Okay, so bottom line is, in the human body, there's a lot of CO2. Okay, so if you haven't done so, you should answer those, guys. Answer them now, so we don't have to do this over the break. Then there's some motility questions. Ooh, anybody remember from the first lecture exam how are bacteria, flagella, how are they structurally different? So guys, sometimes students miss these things that are like bold. Okay, structure is not, for example, on the last notebook grading, pink and purple aren't structures. Some of you know what I'm talking about. Okay, structure, like something. So how are bacteria structurally different? What are bacterial flagella made of? You guys remember this? Flagellin. Somebody has that. Bacterial flagella are made of flagellin. Kind of like Magellan, only flagellin. Okay. Magellan. Oh gosh, that was what, second grade? I can't remember what. It flew around the world. Did you notice that? I don't know where that came from. No, they didn't fly around the world. So not Magellan, but flagellin. So what are eukaryotic flagella made of? Tubulin, which is essentially like microtubules. You guys remember microtubules from, you know, Bio 20, Bio 11? So eukaryotic flagella are way more complicated. They actually have this, you know, wave-like thing going on because they have microtubules that do this cool stuff. It's actually amazing. Whereas bacterial flagella are made of this rigid stuff called flagellin, and they can't do that whole undulating thing. They just kind of turn like a propeller. Okay. Number 2, why don't all bacteria have flagella? Be specific as to their environments. So the big clue is environments.
Ones that live in more liquid environments.
One's correct. Yeah, they live in a liquid environment. Thank you, Jeremy. If they live in a liquid environment, they need to be able to swim around to get to nutrients, to get away from harsh conditions. but the flagella don't help them in a dry setting. It's like having, a fish out of water, that analogy. The fins don't really help them on land. Okay, if you couldn't see them with motility media, what other tests could you do? Anybody remember? Here we go. This goes way back. Let's try that. Hey, what's that? Somebody saying it? Oh, special stains. Thank you very much. Okay, so special stains. You guys remember way back when? I think it was in here. Way back when the first lecture exam was like a nightmare. Hopefully not. But special stains. Way back when, flagella stains. So there's a picture here. This is from the cell growth. So hopefully vaguely remember there's different kind of special stains that are done for special structures like endospores or capsules. We did these two stains in class. We didn't do this one because it's pretty hard. It's even harder than the capsule stain. So flagella stain, you can see the flagella. So that's right, Nima or whoever came up with that over there. Thank you. So, spirochetes, C. tortora, chapter 4. I'm gonna let you do that one. How do spirochetes get around? It's in chapter 4. Okay. So I'll let you kind of figure it out. I can't give you like everything. I've given you pretty much all the answers to these. So fill those out while I go get the next stuff that we're doing. Don't put it off. Please just do it now. Write it up in your notebook so this over the spring break you can enjoy a little. Maybe I gotta work.